Welcome to Chapter 10, Part 2, DNA Structure and Function. Here's where we look at the experiments that identified DNA as the molecule of inheritance. So here's a lovely little timeline. This is DNA discovery timeline. Know the scientists and the discovery. Approximate date is fine, like the 1860s. Both Miescher and Mendel were around. They were contemporaries, 1960s. Miescher discovers nuclein, or DNA, of the nuclei of white blood cells. So basically the dude went to a hospital, collected some pus, and isolated DNA from it. He called it nuclein at the time, this really stringy stuff. Mendel did his work in the 1860s as well, and he published his principles of heredity. His work was rediscovered in 1900. But these are the two you need to know from here. There's other stuff that's going on. There's a bit more you need to know from this timeline, and we're going to talk about all of these that are circled here. So in the 1920s, Griffith demonstrated what's called the transforming principle. In the 1940s, Avery, McLeod, and McCarty demonstrated that the transforming principle that Griffith found was actually DNA. Again in the 1940s, Chargaff and colleagues discovered the regularity in base ratios of DNA, which Watson and Crick are going to use a bit later to discover the secondary structure of DNA in the 1950s. Hershey and Chase are scientists that were also in the 1950s, and they demonstrated that DNA is genetic material in bacteriophages. And also in the 1950s, we had Frankel, Conrad, and Singer show us that some viruses actually use RNA as genetic material. So you should know all of these scientists, the approximate dates, and what they discovered. Now we're going to talk about their experiments, and so you're going to need to know some of those details as well. But what were they looking for? Well, genetic material should have some characteristics, and these are the key characteristics from your textbook. So genetic material possesses several key characteristics. 1, genetic material must contain complex information. 2, genetic material must replicate faithfully. 3, genetic material must encode the phenotype. And 4, genetic material must have the capacity to vary, mutations, recombination, etc., because we want diversity. We want genetic diversity. If everybody was 100% the same, we'd be clones. And that's not healthy, it turns out, because we could all die from COVID, for example. All genetic information is encoded in the structure of DNA or RNA. So we're going to look at Griffith, Avery, McLeod, and McCarty, and Hershey and Chase experiments here. So Griffith, back in the 1920s, specifically 1928, demonstrated transformation in bacteria. So what did he do? Well, he took this poor little mouse here, and he infected that poor little mouse with one of the strains, S-T-R-A-I-N, strain, not strand, like in DNA strand, no, strain. He took an S strain, and it has, it's a diplococci, it's of Streptococcus pneumoniae, which of course gives the poor mouse pneumonia, and the mouse dies. And it has this nice protective coating over the diplococci. Coccus is a form of a bacteria, a shape of bacteria, and diplo means there's two of them that come together. So it's a deadly, lethal form of Streptococcus pneumoniae, and the mouse dies. So he then also took a different strain, S-T-R-A-I-N, of those bacteria, Streptococcus pneumoniae, called the R strain for rough, S stands for smooth because it has this nice lipid coating over it, or nice smooth coating. It's probably what do you call that? Peptidoglycan. R here is rough. So it's not in diplococci format, and it's not a lethal form of the bacteria. So the mouse can be infected with this bacteria and it lives. So he thought, well, I'll do another experiment. I'm going to heat kill the lethal strain. And he did. Boiled it in some, boiled the broth for a bit, like 20 minutes. And it killed all of the bacteria. And he gave the bacteria to the mouse, and the mouse lived. So then he does the fourth experiment. He heat kills the smooth, virulent strain, the lethal strain of the bacteria that did not kill the mouse. And he took the rough strain that did not kill the mouse, and he mixed them together. And then he gave it to the mouse. Mouse dies. He did an autopsy on the mouse, and he recovered live S strain. And that's when he said, something in the dead S mixture, this dead S mixture, transformed, or basically changed, the R strain into S strain. And what was that something? He did not know, so he called it the transforming principle. Well, a few years later, 1940s, I guess about almost 20 years later, Avery, McLeod, and McCarty came along, and there's like, hey, there's this interesting experiment. I wonder what actually transformed these bacteria. So they're asking themselves, what are the possible molecules of inheritance? So they grew up some of this virulent S strain, this pathogenic, this lethal S strain of bacteria. And they didn't kill it. What they did is they let it grow to a point where the cells burst, and then they filtrated off the liquid part and left the live bacteria behind. Just kind of like how you strain your pasta over a strainer and the fluid comes through. This is the fluid, and the noodles stay behind. That's the bacteria staying behind. So only the filtrate, no live bacteria in here. And they're like, okay, well, we know there's three molecules. There's RNA, there's protein, and there's DNA. So one of those is likely to be responsible for transforming or changing the bacteria. You know, it's got some passing on traits, inheritance. So the first thing they did is they destroyed the RNA with an enzyme called RNase. And yes, you should know that for the exam. Everything I'm talking about, you should know for the exam. So RNase destroys RNA. So they got rid of the RNA in this filtrate. They took some rough strain bacteria that don't kill the mice, and they added this filtrate to it. And they asked the question, do we get transformation to S strain? And if we do, we'll see red bacteria down here in this vial. And of course, what do we see but red bacteria? So it transformed the bacteria in the absence of RNA. So by process of elimination, that means that RNA has nothing to do with this transformation because we got rid of it and it's still transformed. So then they got rid of protein using protease, which is an enzyme that's going to destroy protein. They took the rough strain of bacteria again. They exposed it to this filtrate that they treated with protease, and they still got transformation to the S strain. So that meant that protein, when they got rid of protein, that meant that protein had nothing to do with this transformation. So RNA, get rid of it, still get transformation. In the absence of protein, still get transformation. So there's one molecule left, and that was DNA. And so they got rid of DNA using an enzyme called DNA S. It destroyed it. They treated the rough bacterial strain with this filtrate and asked the question, do we get transformation to the lethal, virulent, pathogenic strain? And as you can see from this bottom flask down here, there are no red virulent bacteria, only the rough, non-virulent bacteria. So in the absence of DNA, you get no transformation, which of course means that DNA transformation allows inheritance of traits or phenotype. So DNA is the transforming principle. It's the molecule of inheritance according to this experiment. And that was what they concluded, that DNA is the transforming substance. It suggests that it's genetic material. And it also encodes phenotype. And so this meets one of the criteria for the molecule of inheritance. Well, we don't just do one experiment or two experiments and say we're done. We're scientists and we keep asking questions. So some other scientists came along and they still want to know, is DNA passed on from one generation to the next carrying complex genetic information, or is it protein? So this was a big debate back then because they really didn't know. And Hershey and Chase, scientists named Hershey and Chase came along and they did an experiment. And so the experiment's named after them. And they tested another of the criteria of the molecule of inheritance. They wanted to know, can you pass this trait down? So what they did is they took some bacteria, E. coli, Escherichia coli, that grows in your gut, and they used a virus that infects that bacteria, and that's called a bacteriophage, or phage. I like to say phage, but it looks like this, which looks like an alien from science fiction movies. And of course, science fiction is based on science, and they just put a little fiction in there. But basically what this thing does is it injects its DNA, we now know, into the bacterial cell. And when it does that, this little red phage chromosome makes some proteins and everything. It basically hijacks the cell. It takes over. It's going to break up the bacterial chromosome, and it's going to use the enzymes in the cell, like DNA polymerase and ribosomes and stuff, to make its own pieces, parts. And then it's going to break open that cell, just like Sigourney Weaver when Alien comes out of her belly. In those Alien movies. So this is the lytic life cycle of a T2 bacteriophage, right? So this is pretty complex. You're taking over the machinery of a cell and using it for your own purposes. So this is complex information. So Hershey and Chase wanted to know, can we pass, does DNA do this, or does protein do this complex activity? Okay, well, there's my little bacteriophage causes bacterial cell lysis, which means you break it open or break it to pieces. So Hershey and Chase experiment, 1952, structure of what are called the T-even viruses. They contain DNA inside and then protein in this spaceship looking thing that's really also looks like a hypodermic needle that can inject things into with little legs. Truly alien looking, but it's all protein on the outside and DNA on the inside, and that's what makes up this virus. So Hershey and Chase experiment is shown here. Experiment 1, they take their T2 virus and they infect some Escherichia coli bacteria, and they do that in a flask with some media that lets the bacteria grow. And here's a little magnification of what's happening. They mark the protein with a radioactive material called S or sulfur 35. That's going to label protein but not DNA. For example, cysteine is an amino acid that has sulfur in it. And so after the infection for a while, they interrupt the infection with a blender like you'd find in your kitchen, and then they put it in a centrifuge that has an action very much like your washing machine. And we're going to have centrifugal force that's going to pull down, instead of your heavy clothes, it's going to pull down the heavy bacterial cells, and the water is going to be up top that we can pull off with a micropipette. And what they did was they looked for where did the radioactivity go. And they found that the radioactivity did not go in the cells. It was found in the what we call supernatant or liquid portion of this tube content. And the bacteria themselves in the pellet did not have the radioactivity. So what this says is that the protein is not the thing that got into the cell and causes the change, the transformation, which is the lysis behavior. Okay, so they know it's not protein according to this experiment, but as scientists, we don't stop at just one experiment. We go on and ask the question in a different way. So they again took this virus and the bacteria, and they let the virus infect the bacteria for a while. This time they're using a virus that's labeled with phosphorus 32. This is a radioactive isotope of phosphorus, or P32. They interrupt the infection using your kitchen blender, and it just shears those virus particles off of that off of that cell. And then into the centrifuge, much like your washing machine, and the heavy bacteria pellet to the bottom, and the liquid stays up top. And they asked, where is the radioactive material? And this time they found that the P32, which was labeling the DNA, because DNA contains phosphorus in the phosphate group, and proteins do not contain phosphorus. So they knew that what got into the cell and allowed for this big change where the cell was converted into a virus factory was due to DNA getting in there. So that suggested, P32 DNA found in the bacterial pellet. So this suggested that DNA is being passed from one generation to the next, carrying complex information such as the ability to lyse a bacterial cell. And so DNA meets another criteria for molecule of inheritance. Concept check. If Avery, McLeod, and McCarty had found that samples of heat-killed bacteria treated with RNase and DNase to get rid of RNA and get rid of DNA transformed the bacteria, but samples treated with protease did not, what conclusion would they have made? So I told you needed to know those enzymes. A, protease carries out transformation. B, RNA and DNA are the genetic material. Nope, no transformation happened when we got rid of them. C, protein is the genetic material. That sounds like the right answer, but let's read this last one to make sure. D, RNAs and DNAs are necessary for transformation. Nope, that's not true. So C is the answer. Protein is the genetic material according to this experiment. And this is just a what if. Okay, this isn't reality, but this is a way we can ask a question to see if you understand what happened in the experiment. So you should know how to answer a question like this. Okay? All right, experiments to discover DNA secondary structure included Watson and Crick's discovery of the three-dimensional structure of DNA using Chargaff observations and Franklin data. And her data was the X-ray diffraction image of DNA. So Chargaff and colleagues in the 1940s did some sampling of different sorts of species and looking at the DNA in each one of those. And they created this table, base composition or percent of DNA from different sources and ratio of bases. And that sounds very exciting, I'm sure. Well, anyway, so they looked at the A's, the T's, and the G's, and the C's, and the ratio in which each species had them. So E. coli, has 26% A, 24% T, 24% G, 25% C, and then they found the ratio of A to T was approximately 1, and the ratio of G to C was approximately 1. And if you added A to G, the numerators together, and you divided them by the denominators T and C added together, that would give you a ratio of approximately 1. And that wasn't just true for bacteria, but for all of the species they looked at: E. coli, yeast, sea urchins, rats, humans. So that was the data that was really interesting to Watson and Crick. You can see also, just, by the way, E. coli and yeast have very different contents, And that's part of what makes one species different from another, is the genetic material ratios between the nucleotides. But anyway, this part right here, the almost, ratio of 1 to 1 was what was really interesting to Watson and Crick. Now, also what was interesting to Watson and Crick was Rosalind Franklin's data. And here's Rosalind here. She was at King's College in London in a lab of Maurice Wilkins. And she was an X-ray crystallographer. Watson and Crick used her data. She did not know that they looked at her data. Wilkins gave them permission to look at her data. She was not present at the time, which is a bit shady. But, she was a woman, so. Anyway, don't get me started on that. She died in 1958 of complications from ovarian cancer, probably because she used all that X-ray to look at DNA structure, but we don't know. This is what she did. This is Franklin's X-ray crystallography data. So she would precipitate the DNA molecules into kind of a salt molecule, and it would be in a crystal. Salt's a crystal. But she would precipitate it, and she would get a crystal structure for the DNA, and then she would bombard it with X-rays, and then it would give a diffraction pattern, similar to what we get when, we take an X-ray of our bones, for example, to see if they're broken or whatever. And I don't know anything about reading a diffraction pattern, but when Watson and Crick looked at this and other folks, it looked like there was a pattern that repeated over and over and over again for DNA. And to someone who knows what they're looking at, apparently this looks like a helix pattern. So that gave Francis Crick, who was actually a physicist, and James Watson, who's a biologist, the idea to make this model of a double helix that they published in 1953. And it included a purine across from a pyrimidine, such as Chargaff's data suggested, and it included this double helix as Franklin's data indicated. So, can DNA replicate faithfully? Well, we haven't answered that question quite yet. We'll get to that in another chapter. But Watson and Crick did have, in their very small paper, no figures in it, mind you, in Nature of all places, which is a very prominent journal today, and you have to have many, many, many data figures in order to get published in Nature these days. But they had like just over a page of written text, and it was published in Nature. And the title of the paper was A Structure of Deoxyribose Nucleic Acid by James Watson and Francis Crick. And in the paper they had this lovely quote. And I can't do a British accent or anything, but it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. In other words, they saw this hydrogen base pairing between the purines and the pyrimidines, and they knew that those were easy bonds to break. And so they knew that there was possibly a way for this molecule to replicate itself, which was an important feature for the molecule of inheritance. So this suggests that DNA has the structure to faithfully replicate. That still needs to be tested, and we will be looking at that experiment in another lecture. This may meet another criteria for the molecule of inheritance, and we'll be looking at that in chapter 12. So for their work, these three dudes got the Nobel Prize in Physiology or Medicine in 1962. Maurice Wilkins, this is the guy who let Watson and Crick look at Rosalind Franklin's data. She would have gotten the Nobel Prize too, except she had died of ovarian cancer before it was awarded. All genetic information is encoded in the structure of either DNA or RNA. And here we're going to look at tobacco mosaic virus to show that RNA can also be carry genetic information. And notice we're looking at a virus. So the viruses in this diagram are color-coded. There are two different types. We've got one color-coded yellow, the other color-coded purple. And inside so this is the protein coat. Looks like kind of corn on the cob here. And inside is RNA in this helical structure. And we've color-coded this other strain of the virus, purple, the RNA inside, and the proteins on the outside. So what Frankel Conrad and Singer did in 1956 was to create a hybrid virus, shown here on the diagram. And inside was the HRV RNA, so one type of viral RNA was on the inside, and on the outside was the proteins from the TMV virus. And then they infected a tobacco leaf, and they looked at a lesion, and they asked the question, what does it look like? Well, it looked like the virus, as you can see very easily on this diagram. It looks like the HRV type or the yellow type. So whatever the nucleic acid was, that determined the type of virus that was the genetic information that was passed down. So this suggested that, again, it was not protein, but nucleic acid that carried genetic information. The progeny from hybrid virus will look like the virus from which the RNA was taken. Therefore, RNA is the genetic material in these tobacco plant viruses. Okay, so that's from your textbook. RNA can be genetic material. Tobacco mosaic virus, TMV for short, Frankel Conrad and Singer. Okay, so in summary, it took many scientists and many experiments over 100 years to work out the secrets of heredity. And we're still learning more every day. That's why my genetics textbook keeps changing every couple years. All right, well, that's it for today. Next time we'll talk about something else. I know you're looking forward to it.